Hi, Dr. Jen Vitry here with Vitry Wellness. Today we're going to talk about Nutrigenomics, unlocking personalized nutrition through genetics. Many practitioners focus on a single gene variant. I'm sure you've heard of MTHFR gene or the COMT gene. This approach overlooks the complexity of gene interactions with biological pathways. Their effects depend on interactions with other variants. It's sort of an interconnected highway. A broader, integrative genetic assessment is essential for effective personalized diet and lifestyle care. Relying solely on an isolated gene can lead to incomplete or inaccurate guidance. Multiple gene variants interact dynamically within shared biological pathways, influencing health outcomes beyond isolated effects. Pathways such as methylation collectively mediate nutrient metabolism, amplifying or mitigating individual gene effects. Integrating gene variant interactions ensures recommendations reflect genetic complexity, optimizing individual health strategies. Assessing gene groups rather than single SNPs, single nucleotide polymorphisms, also called SNPs, improves prediction of biochemical outcomes and tailored health interventions. So integrating genetic testing into holistic patient care. This is something that I do in my practice, and I'm going to walk you through a little bit of what that means, how we look at it, and then talk about a specific report. There are a number of reports you can run, but there's one specific one that I really enjoy, and I'll explain that to you. But genetic testing as a complementary tool, it supports but does not replace clinical assessments and identifies genetic predispositions influencing health. Incorporating lifestyle and nutrition helps us to really evaluate diet, exercise, and habits that are impacting health and addresses modifiable factors alongside genetic risks. While assessing environmental exposures is very important, we need to consider pollution, toxins, living conditions, and links to environmental gene expression and health outcomes. And I'm sure if you've reviewed the 47 steps, you will see that there's an exhaustive list and many videos covering so many environmental toxins and how to address them. Integrating laboratory and clinical tests includes blood work, biomarkers, diagnostic results, and correlates lab findings with genetic and lifestyle data. All of this is helpful when looking at the genetics. In enabling personal treatment plans, you take all of that data and you tailor interventions and maximize the potential for improved patient outcomes. Now, genetic variation shapes individual health. Nucleotide substitutions alter single DNA base, impacting traits like hair color and stress response. Deletions and insertions add or remove a DNA segment, which is potentially changing like protein function and metabolism. Copy number variations involve changes in DNA segment repeats, which influences nutrient metabolism specific and disease risk, for instance. And genetic variation accounts for only about 0.1% difference in individual DNA, underlying unique health traits. Understanding these variations enables tailored lifestyle and nutrition recommendations for optimized health outcomes. So Nutrigenomics is personalized nutrition through genetics, essentially, and we explore how genetic variations influence your individual responses to nutrition and diet, which is very amazing. Focuses on gene environmental interactions driving the gene expression. We'll talk a little bit more about that, which predicts health outcomes. Enables tailored dietary and lifestyle recommendations beyond one-size-fits-all advice, which is super important to me because this is an individualized science-based approach to health and wellness based in nutrition. I'm a nutritionist, functional nutritionist, and it allows for individuals to support pathways with greater precision and shorter time span. We can be effective and efficient. So how do we really tell the difference what makes us vulnerable when we're looking at the DNA code if there's really only 0.1% difference? And this is when we talk about SNPs, which is the single nucleotide polymorphisms at a specific location, at an allele that allows your genes to predict sort of a more at-risk variant. When we look at the SNPs and we see that in your location you have, say, AA instead of AT. AT is the protective pathway or the upregulated gene. You have AA, so you have more of a downregulated gene there. That SNPs is very common genetic variations occurring in coding or regulatory regions. SNPs can influence gene function, expression. It can impact biological traits, and obviously, if it's a risk allele, it influences disease risk. And that's what we always hear about. We always hear about you have this gene, this SNP, and therefore you are at greater risk for this disease. The way we're thinking about it is we're going to look to see where the SNP is located, and we're going to group them up, and we're going to try to figure out what nutrients would help support that SNP so it's more upregulated in certain body pathways, and we'll talk more about that. But genes are represented by capital symbols, while protein products are denoted by lowercase notations, reflecting the translated protein. Sometimes it's important to kind of know this notation so that you can kind of interpret it with more clarity. Okay, so pathway analysis. Clusters of low penetrance genes are analyzed together rather than individually to assess their cumulative impact on health. Genes interact with biological pathways, reflecting the complex relationship that contributes to multifactorial traits. This approach improves understanding of genetics susceptibility, supporting more precise and personalized health strategies. So here's a good example. If we take particular SNPs that are triggering, see if I can blow this up a little bit. So in this area right here, in the top left, you can see a few SNPs, and you'll see with the pictures next to it, those SNPs are particularly sensitive to environmental factors, and then you combine them with the SNPs below that are sensitive to physiological inflammation, then we can sort of assess, okay, this person has the ones that are triggered by this and sensitive to this, so the cluster of these SNPs combined make this person very vulnerable to chronic inflammation. That's what this diagram sort of shows, and how we think about it, how we pull together the different genes to create a story. And I want to help counteract that story with nutrients and life. Here's another example looking at impaired cognition, for instance. Think about processing speed, we think about working memory. So here we're pooling potential downregulated genes, all of these genes in the second column that have to do with cognition, to assess potentially impaired mechanisms and the accompanying biomarkers. You probably see a few genes you've heard of before, like the MTHFR gene that deals with methylation. They are part of many symptom assessments as they turn other genes on and off. And you can see with this third column and the fourth column how we're taking the SNPs, deducing what could be downregulated, and thinking about the biochemical pathways and the outputs of this situation. So now we're going to talk a little bit about epigenetics, part 2, which is the how to quiet the genes through lifestyle and diet. Transitioning from genetic variation to gene expression control, this section explores how epigenetic mechanisms actually influence the health outcomes. We'll delve into the dynamic interplay of lifestyle factors and gene activity, setting the stage for understanding DNA methylation and its role in personalized nutrition. So looking at how nutrients modulate metabolic pathways to influence cellular health is an amazing process. And there are many reports that walk us through it, but this is my current, this is the one I use the most, and I'm going to show you a number of reasons why. Most importantly, it allows the client to walk with me through the process. There's a lot of graphics, there's color coordination, and it helps my client understand why I'm targeting certain areas to emphasize over other. On your left side here, I know it's pretty small. I tried to capture a whole page. But so in the beginning of the report, you get your gene results, and this is typical. Here's protective, slow response, no impact. And there's a lot of no impact. So just remember that it's not all bad news. Any information is good information is how I think about it. But if you look on the right, they take the genes that are impacted slightly or very much, and they color coordinate them, and they group them into various system functions. This is particularly helpful with trying to navigate how to prioritize your health strategy. So it helps us to determine the stronger or weaker system functions and where we should focus our energy. You can see through color coordination from indigo to purple, and then there's blue, and then finally yellow. What is the priority? So yellow needs the least support, and blue, and then of course indigo at the top there demands the very high or most attention. And the whole report, I can't go back a slide, but as you know, each system, as I showed you in the last slide, outlines the different colors, and it's immediately noticeable what areas are going to need the most attention. The report goes on to show graphics about the different systems so people can understand them, mitochondria, the impacts of oxidative stress here, detoxification, and I can walk the clients through the physiological and cellular expression pictures so that they can sort of understand what is oxidative stress and how is it impacting me, how is it creating inflammation, how do we get it out of the body. Here, this person is great on detoxification, but as it's purple, so we need more attention with oxidative stress. Here's another graphic. Methylation, we were talking a little bit about this before with the MTHFR gene. There's two main mutations that are considered, and they're either heterozygous or homozygous, meaning you get them from one parent or two parents. And remember, just because you have a SNP, like I'd say, I forget the recent statistics, but 60% of the population, 60-65, have at least one of the two variants heterozygous. And just because you have a SNP doesn't necessarily mean it's impacting you. Usually if it's homozygous from two parents, you have a greater potential for it not to do its job well in your body. So methylation is stage 2 detox. It's also mental health. It's where you take your folate and methylate it and turn it into B12 and methylate it. And the most important thing right here is that homocysteine then becomes methionine, which then goes on to the next pathway. Homocysteine is inflammatory. So methylation also turns genes on and off. So it's super important, super powerful. It's something we want to figure out, and this test looks at it closely. You can see in the graphic here that they talk, and it's just 1, 2, and then there's 3, 4 at the bottom, because they really want to explore your methylation pathways and which aspects of it are working or not working, according to not just the MTHFR genes, but other genes in the cluster. And then this report here demonstrates, I really like this because on the left here, it demonstrates for the client what each aspect of the various system assessment is doing with great graphics. So it walks them through each step of the energy overview and how the different SNPs correlate with each aspect of it. And of course, on the right, it does a good job portraying nutrient needs and highlighting which one is most effective and necessary. In epigenetics, this is how diet, stress, and environmental dynamics regulate gene expression to impact health. It's right. Epigenetics is gene regulation through diet and lifestyle, because essentially we can quiet the genes, the SNPs, if we provide the support for those genes, which is diet and lifestyle. So nutritional genomics, explore how genetic profiles influence individual responses to diet and nutrient metabolism. Utilize personalized nutrition strategies that integrate genetics and lifestyle factors for a tailored health plan. Support custom interventions that improve nutrient absorption, lower disease risk, and enhance overall wellness, and drive better health outcomes by aligning diet and genetic predispositions and environmental influences. Just think about it like health equals genetics plus choices, because it does shape your health. Here's my references. And thank you so much. Reach out if you have any questions, and I look forward to seeing you in Healing ALS community and their groups and chats and further workshops together. Take good care.